Okay, so welcome everybody to the new Ipanema Scientific Online lecture. This is going to be a lecture on capillary microfluidic chip design, so very relevant to Ipanema topics. And our speaker is Guillermo Artana from Argentina, from the CONICET Institute and the University of Buenos Aires. And Guillermo, the floor is yours, and let's hear. Thank you.
Well, thank you very much. I'm pretty glad to present some of the results we have obtained and some contribution that we can share with you related to this kind of topics. So my laboratory is in Department of Mechanical Engineering, and it's related to fluid dynamics. So the main aspect of this topic is related to fluid dynamics aspects of capillary microfluidic. So a part of my lab is, or large part of my lab is related to flow control. And in microfluidics flow, you have also flow control, or a large part is related to flow control. So we divide usually flow control into two main branches. One is related to active flow control and the other is related to passive flow control. The difference between both of them is that in one case, in active flow control, you incorporate energy to the system. On the other hand, in passive flow control, you don't need to incorporate any kind of energy. So one possibility that you have with active flow control is that you can propose some kind of tuning of the results in function, as a function of the goal you want. That means that you can propose closed loop systems. On the contrary, in passive flow control, you cannot undertake this kind of procedure. So among the different possibilities, there are different possibilities of active flow control. You can force the flow with pressure gradients or with electrohydrodynamic force. Here you have a video in which you can see the effect of electrohydrodynamics. That means that you incorporate to the fluid an electric force, or you can also tilt the platform and produce take advantage of gravity force. So there we're tilting the platform, and you can see in a junction that is here the movement of fluids on one sense and on the other. Well, this kind of topics were developed related to some of the contacts that we have with industry. Most of them are related with startups. But some time ago, we decided to go to another aspect that this was related to, well, passive flow control and to take advantage for capillary forces. And there appear two main systems, one that is paper-based and the other that would name channel networks, that in which it is not based on paper-based, but that you construct and you build channels networks in a chip. So my topic is mainly related to channel networks, but I will also talk about some aspects of capillary forces and how we use them. So why we have tried to move a little bit to this kind of passive flow control? Well, active flow control is quite interesting, and you can do a lot of things, but usually you need of highly experienced users. And in some cases, instead, initially microfluidics appear like as a system that was named lab on a chip or something like this, but some people started to call it lab around the chip because there were a large amount of peripheral equipment that require necessary of highly experienced users. So at the same time, you have a lot of complexity, and these kind of devices are interested to undertake research, but they are quite difficult to scale up. So there, microfluidics, in my opinion, enable a lot of research for biologists and biochemicals, but there was a little bit of difficulty to scale up and to pass to a large aspect of industry. As we are engineers, we are interested to this kind of transfer to the industry. So one of the reasons we did not abandon the other aspect, but this was one of the reasons why we were interested to go to capillary microfluidics. So in general, in this process, you have different stages: chip design, channel fabrication, surface functionalization, readout devices, and mobile app telephones. And in passive flow control devices, what we are now more thinking that we can help some industry and some enterprises to produce our point-of-care testings, home medical testings, some applications that are related to low sample tests, like for small animal kits and so on, and also for field tests when you don't have all the equipment you require or that you may have at the lab. So our expertise is more related to flow control, but well, they are, as we are engineer, also we have in our in our projects or in our conception of the systems, also present the manufacturing engineer and instrumentation. So what are some of the advantages of capillary microfluidic systems is that they work with no peripherals, they are user-friendly operation, they are easy scale-up production and low-cost final products. What we can say that they have as pros and cons of capillary microfluidics. Pros, well, they reduce costs and complexities. You don't need any pump, connectors, monitoring system, and so on. They are quite easy to use for people not very experienced. They are compatible with also with people that work in the labs that are used with pipettes and so on, and then they are well adapted to diagnosis. On the contrary, there is a problem that you have no feedback mechanism to adjust operations, so you have to, once you design it, you cannot tune any variable. So you need of a careful chip design. They are quite limited to use in cell culture. This has been some aspects of that have enabled development of microfluidics, and also they do not allow the possibility of some recirculating flow that can be of interest for biologists. So in this talk, I will be talking mainly of capillary networks, and I have mentioned in the first one in the second slide something that we have also some neighbor successful technologies. They are paper chips. Well, we have in Ipanema consortium, we have people from China. I think the name, I apologize if I don't say it correctly. I think is Xi'an Xiaotong University, that are specialists of this kind of devices. And I just want to mention, well, some successful application of this kind of devices. One of them is the well-known home pregnancy test. And the other one is, well, there are many, but I just mentioned two of them. The other one was related to COVID and the possibility to detect the presence of COVID in people. So the the main aspect in paper-based is that you have a pad and you put the sample in in a reservoir, and then it flows through capillary forces. Now there you don't have channels. Actually, what you have is a matrix of cellulose or nitrocellulose that may be accompanied by some hydrophobic barriers. So in the case, when should we consider capillary microfluidic chips instead of traditional paper-based devices? Or what will the advantage of capillary microfluidic chips against traditional paper-based devices? Well, I would say that they are not in opposition, but they are complementary. But one of the aspects that I think that we have to highlight is that the possibility of flow control is more reduced in the case of paper-based. On the contrary, in the case of capillary microfluidic chips in which you construct your channels, where you have a large possibility for, you can have access to more design tools. So as in this case, also in capillary microfluidic chips, you can manage, because you have larger possibility to control the flow of time of the process. And also you can control more the shear stresses that will be subjected or that will be imposed to the sample. Also, you need, in general, for capillary microfluidic chips, low sample consumption and also of reagents, and you can have the possibility to have transparency on the chip. This means that you can use a large number of tools related to optical detection. At the same time, I would mention that it's quite well adapted to most physiological fluids that are in general non-Newtonian and rather viscous, or maybe rather viscous. And I think that I mentioned that they are well adapted to most of them, because you don't need to condition, to make conditionings of the sample, or to treat the sample, or to handle the sample prior to use in the traditional paper-based devices, like in the paper-based devices. Also, it is quite simple to propose multi-target detections. This is possible also in traditional paper-based devices, but have some complexities. So I think that there's some possibilities that are quite interesting for capillary networks that should be or can be exploited, and that's the reason why we have some time ago started to work with this kind of devices. Well, we are. You may have two different systems or you may classify the capillary system or networks into two large groups. One group is in which you have closed, the flow is in closed channels, and there's another one in which you have also there's no cover, and you can have an open channel for flow. What I will refer are those related to in which you have a closed channel flow. So in this kind of devices, or most of the devices that have been developed, you have a pump here, or a region in which you increase the surface, and the system is controlled, the flow is controlled from the loading pad. Here you have the reaction chamber through a flow resistance or a system that is quite intricated, and that allows to reduce the flow through the imposition of a flow resistance that is the limiting resistance of the system. So in this kind of devices, what you usually have is a kind of constant flow devices. But, well, there may be some interest, or there may be larger possibilities if you can pass to another system in which you can have a larger control of the flow and of the amount of liquid a long time that you are delivering. So these systems, the system that we propose, or we are interested in, is to develop systems in which you can have a large control of the flow rate or of the volume a long time. So that's the reason why we have abandoned a little bit the idea of limiting resistance, and try to have a larger pools of designs based on different aspects of the chip, or on the channels, on the flow, that I will some of them mention today. So just to mention, you can have different control of flow rates a long times in this range, from microliters per seconds to nanoliters per seconds. This means that you can have very low flow rate control. You can also have a control of the volume delivered a long time in the range of microliters to nanoliter, and also that you can have a share stress control, or you can be sure of the stresses that are the sample being subjected and they are in the range of Pascal to millipascal. And well, just for people that work on organ-on-a-chip, 1 millipascal is about 0.01 DINs per square centimeter, and usually people in organ-on-a-chip work with within this range. So these chips are usually accompanied by, or can be accompanied by some valves, and you can have valves that in which you stop the flow, and you can have a break of the process there, or you can delay you can have what we name as delay valves, in which you stop the flow, but the flow goes on some time after you produce the stop of the flow. So I will mention one of them here. In this video, we can see a stop valve here, and how the process is recovered once it passes another flow there. So we put 3 liquids there of different colors. These are the channels that are there, and then you may have this, and then there will be a flow coming from the other side, and flow starts from the other 3 that were on the right. So these kind of devices you can well mix. This can be used for mixing or whatever you want. In any case, this is one of the possibilities, but also you can have other possibilities in which you take advantage of dead gas volumes. So you can propose valves that once you release the trapped gas, you can produce the flow. So this is one of the examples And when remove this cover there, well, you can produce the flow. Also here, there's another example. You can produce different gradients of the fluids, and then so well, these are possibilities that you can go ahead with this kind of devices. Obviously, we are not the only people that are working in this kind of developments, and there are some companies that have already started to work with this and or are having some experience with this, and I mentioned some of them here. This is a consortium also from the Europe that is a very large one. So there are some aspects also that appear in the design as we don't have any tuning parameter. What we have to be quite sure is about the design and that the correct design of the chip, so that it produces or it happens what we wish that occur. So one of the problems that appear with this kind of devices is that you may have asymmetric feelings. You may go like this, and then you see there that this branch is going a little bit slower than the other one, and this is produced exactly here. So these were symmetric bifurcation, and however, you don't have this kind of symmetry. So you have to be, there are some tips or some aspect that we have been studying concerning to T-junctions and curves and this kind of singularities, so the fluid goes as we wish. The other aspect is that the flow has some chlorine aspect. It doesn't. If you have a look here, you see some chlorine of the advance of the fluid or the filling of the capillary. And also another aspect that is of interest is that this is the front here is filling, and you will see that in some cases, and depending on the aspect ratio, you may have some fingers that goes along the laterals of the corners of the channel, and this is called concus fin movement. So here we see this is the front of the meniscus, and you see also that you are having some precursors that are going quite ahead from the meniscus. So well, these are some aspects that have to be taken into account in capillary chip designs. And well, I will discuss some of these aspects in this talk. So first, I will give some in the rest of the talk. I will first give a brief description of chip constructive method that we are using, very brief, so you understand why we have undertaken some studies to have a larger control of this kind of devices. I will explain a little bit of capillary pumping design. I will show some modeling of filling stacked capillary channels, well, a description of a code that can be used from anyone for this kind of devices, and then how we proceed to have strategies related to some objectives that we may have, and these are produced with genetic algorithm or machine learning genetic algorithm. So some of the these two aspects are in our paper of 2022 in Langmuir, and this one is in Lab on a Chip of 2022. So more details will be there. And this machine learning is related, has been developed by the first author of this book is Thomas Durier, that he's from our lab and works with us. So in concerning channel fabrication techniques, we use very simple techniques. We make a sandwich with biocompatible thin films that are of the order of 100 micrometers, and we bond them thermally or with adhesives to transparent plates that can be glass or PMMA, or also films. And the channel geometry are obtained cutting patterns on the film through geography. This means with razor blades, a system that is already, it's like a printer that instead of inkjets, you have razors there, or also by system of laser ablation. They also use, you use CAD files or in which you design the film, and then you can produce, and you can cut the pattern on thin films quite easily, and then you bond them to the microscope slides, or to the PMMA that you have already done. So if you use the system with adhesives, you have two liners, and you remove the liners, and you put them inside between two microscope slides, and you can put here your sample. So one aspect that you have here is that you will have two materials in the channel. You will have glass and glass on the top and on the bottom, and in the laterals, you will have a film of a polymer that is biocompatible. So the other aspect that you will have there is that the top and the bottom will be quite smooth. On the contrary, the lateral of the films, you may have some rugosity, and depending on the process you have used and on the characteristics of the film with which you produce the film, with which you produce the channels, you may have different patterns of rugosities, and you may have more rugose or less rugose lateral walls. So you will have two different materials, and glass or the film that is polymeric, and also you may have two different rugosities in top and wall and in the laterals. So concerning the basic equations that we should use, or we can use to design these chips. Well, first, most of them are derived from classical fluid mechanics, in which we express mass conservation, and as we are we can apply in this kind of problems the incompressibility condition. Applying the incompressibility conditions means that the volume is preserving, and so divergence is null. The other aspect is that we apply linear momentum conservation, and this means that you can link the acceleration of each fluid particle to a divergence of a tensor, of the stress tensor, in which you have a part that depends on pressure, but a part that depends on the deformation tube. And this is the viscosity. But this equation applies only to Newtonian fluids. The third equation that you can use is the angular momentum equation, in which you have this one only helps or to illustrate the case that this tensor is symmetric. And last equation is actually is not in many cases required, because with these two equations you have the system closed. You can resolve the full differential equations. So the main aspect is that you have to usually solve these equations in which you have different variables accompanied by divergence null of the velocity fields. Well, if you take this equation and you consider channels, you can pass to, you consider quasi-unidirectional flows. You can link the pressure gradients with the velocities. So here you have, in this table, you have the different geometries, the different ratio between the gradient of pressure and velocity. So for circular channel flows or circular tubes, you have this kind of relation. Here mu is the viscosity, this U bar means the mean velocity, and R is the radius of the pipe. Well, actually, what we use is a system in which you have a square or rectangular section. So here appears C, one of the laterals, and you have here a function of this has to be corrected by a function that depends on C, and the ratio of C, the aspect ratio, C over B. And if you go to a porous media, this is a photo of a scan photo, microphotography of the paper that are used generally in paper-based systems. You have here a relationship to the Darcy law that relates the pressure gradient with the local velocity, or the velocity in the direction of the pressure gradients. You have the viscosity too, and you also have here the two parameters that are related to the structure of the matrix. One of them is related to the porosity, and the other is to the permeability. This permeability is considered in this expression as a constant, but actually this is a tensor too. So when you are in this situation, you have expression, you can know which is the local velocity field. In this kind of matrix, well, how is the local velocity field is not possible, because this equation is derived from average Navier-Stokes equation. These are directly, these both equations satisfies the full Navier-Stokes equation, but to obtain this kind of equation, well, you have to average on the space, and there are some aspects that have to be satisfied. Well, meanwhile, in this kind of problems, these expressions are related to unidirectional flow, and this means that the flow is follow or is parallel to the axis of the channel, and you are in steady flow systems. In this one, well, you have some aspects that should be satisfied to accept this kind of equation. Well, also you have the condition that you are in steady flows, but this is obtained from an homogeneous isotropic matrix of incompressible and saturated mediums. This means that you have no air pockets. This applies only to low Reynolds number, and also you require that there should be no chemical reaction or matrix dissolving or particle transport inside there. Well, one of the problems that you can observe is that this is the thickness of the paper, and well, this is quite different from the situation that you have here. The other problems that you have is that when you use this expression, is considers the viscosity as a constant, but viscosity may depend on the the share ratio of the problem. That is, it may depends on the deformation of the particles. And we follow here, we fall in the condition that you have, or you are in the situation you have non-Newtonian fluids. So for these two, you can obtain an analytical solution, or you can easily integrate them. And for this one, well, you have no clear expression for non-Newtonian fluids. So many biological fluids used in point of care are Newtonian. Blood, saliva, lacrimal fluid, semen, and so on are all non-Newtonian. And you can dilute your sample, but the relation of the rheological curves with solid concentration or temperatures are quite complex. So there's some limitation in the use of this kind of fluids when you consider non-Newtonian fluid and paper-based devices. So one of the aspects that when you are facing problems in which you have non-Newtonian fluids is that you should go to analytical solution or numerical methods. These are allowed more for channels in which you know the geometry and you can integrate the equation easily. Well, when the whole system of equation that I have mentioned of conservation equation, this means that the equation that are related to mass conservation and momentum conservation can be, you can integrate this kind of problems, and when it should be necessary to have a look to a whole system, or what we mean as computational fluid dynamics. Well, when you are in situation in which you don't have unidirectional flows, you cannot use any longer the equation that I have shown in the previous slide. So these are cases of a mixer or cases in which you are focusing fluids that you cannot use the equations based on lamp parameters, and you have to go to CFD. Well, also there are, besides the aspects or the aspects that are related to bulk equation that I have mentioned previously, you have situations you have some additional questions that you have to take into account. One of them is that you have to consider the boundary conditions. Usually in fluid mechanics, we use the Newton-Meyer principle. That means that we accept the non-slip or continuity of the velocity between an interface and the fluid. But also, and this is one of the aspects in which appears as the main topic in this talk, is that surface tension effects. So there you have a Laplace equation in which you have, this means that if you have a curved surface, you have a jump pressure along them, and this jump pressure is related to the surface tension, and this inverse, this is related to the principal radius of curvature of the interface. And also the other aspect that appears is that you may have a wall, and when you have a wall fluid and air, you will have a contact angle appearing that has some very important aspects in the problem that we are dealing with. So let's first talk a little bit of filling of dynamics of filling of straight channel. This has been described there are some non-dimensional numbers that are of interest in this kind of problems in capillary microfluidics. The first aspect is the first non-dimensional number of interest is the Reynolds number. This is the ratio between the inertial to viscous forces, and in the cases we are looking are usually of the order of 1. The other aspect is related to the Strual number. This is the ratio between the convective and the local acceleration term or inertial forces. In our cases are of the order of 0.1. The capillary number is the ratio between the viscous forces and the surface tension forces, and the Weber number is the ratio between the inertial forces and the surface tension forces. So in stacked channels, usually what you have, or the equation that governs the filling of the channel is described by the Lucas-Washburn equation. This equation is derived from the momentum conservation equation. And here you have the inertial terms. These are the capillary terms. These are gravity terms. In our case, this will be neglected. This is the shunt pressure that you have between this part and this one. This means if you have a different pressure between the reservoir and the head of the channel where you have the meniscus, this term appears. Otherwise, in general, this term is null, as you have the same pressure in the reservoir and here, and also you have the viscous terms. But concerning the real pressure that you have in the inlet, there's a discussion there, but I will not go, or I will not give too many details here. But what is important here to look is that you have a different pressures related to capillary between this point and this one. Here you have a jump of pressure, and so you have a pressure difference between here and here. So you have a pressure gradient, and there's a competition between this pressure gradient that tries to drive the flow and with the viscous flows, the viscous forces that you have in the walls. So this is the competition that you have. And what you observe is that as you increase the length of the that is filled by the liquid, this jump of pressure is related to the curvature of the meniscus. This is constant. As you increase this length L, this pressure gradient between this point and this one will diminish. And at the same time, what you have is that the flow rate is related to the speed or the velocity in which this front moves. This is related to the derivative of L against T, and is proportional to the gradient of pressure, this one. So you can integrate this equation, and what you obtain is that the L, or the filling length, is proportional to the time, the square root of the time, or also what you obtain is the flow rate is proportional to the square root of the time. Now, this equation is derived under several assumptions. The most important is that you neglect the inertial terms. So you are assuming that Reynolds is quite close to 0 and that the Strual number is quite close to 0. Well, if we go now to the problem that we are dealing with, this is the problem of you have one slide here, the other slide here of glass, and you have the laterals here that are of different materials. Well, first, from a static point of view, you may have a a solution of this kind of surface, or the surface of the meniscus that is being formed. This is numerical. This is not quite difficult to incorporate the different materials here. Now, incorporating the shape of the meniscus, and in consequence the pressure difference is, you can obtain it quite easily, but there appear some aspects that complicate or makes more complex the problems. The first one is that you may have rugosity in these walls, and the contact angle will change depending on which part of the pattern or rugosity you may have. So this explains a little bit the growing that we have observed in the first aspect. So one of the aspects that we were interested in was to incorporate in the filling of the channels the rugosity effect. The other aspect that appears is also that this is the numerical, the top view of the meniscus that you will obtain or is predicted by a numerical approach, but you have also the different results depending on if you pre-wet the channel or not. So there's an aspect, if you have small, if you have previously passed a little bit, or if you suspect that there's a thin existing on the liquid film existing on the channel, you may have different results of the meniscus. This is another aspect, and another aspect that I would like to mention, another aspect that I would like to mention is that this contact angle is actually a contact angle in a static condition. And when you are in dynamic condition, appears well, what is called a dynamic constant contact angle. Sorry. So why it is important, the aspect of pre-wetting or not? Well, I have mentioned previously that we have the Newton-Meyer principle that establishes that you have the same velocity of the liquid and of the wall. There is not discontinuity in the velocity at the wall. Where if you consider this condition of this principle, the contact line should not move because the wall doesn't move, and then you cannot accept that the contact line should go ahead or would produce the movement of the fluid. So there's a singularity. This is a problem that has also been or has a lot of people has been working with this, and well, it appears there are different models to take into account this. One of them is the hydrodynamic model that accepts directly that you may have slip condition at the wall, and then you form a kind of precursor here that enables the movement of the fluid. So in this kind of systems, they make they link the static angle, the static contact angle, with the dynamic contact angle through the capillary a dimensional number, non-dimensional number in this way. On the contrary, you may also try to describe the dynamics of the contact line through another model that is related or is inspired through the kinetic theory of model, is named like this, and in which you have a relationship between the cosinus of the static angle and dynamic angle of movement. The contact angle will be so different in the static case and when you have movement, and these are related through the capillary number, and this is a coefficient beta that is named as friction factor. So the main aspect is that you can relate these two, the static and dynamic constant angle, in different ways, depending on the model that you are accepting. They are related through the capillary numbers, but well, in what it follows, we adopted this kind of models, and the problem is that in this kind of problems, you have this, it is not inclusive the rugosity. So what was our first contribution, or the main aspect that we have tried to determine here is, well, related to the problems we were trying to study. We first consider or try to incorporate to the Lucas-Washburn equation the fact that we have two different wall materials, the fact that we have a movement of the line, and incorporated also the possibility of rugosity. So if you combine all these kind of things, the equation that you achieve is an equation in which you have a friction factor, an effective friction factor, and an effective dynamic or contact angle. When you incorporate this in the conservation equation, you achieve an equation that relates the dynamics of the front to these two coefficients that appear here. And then instead of having an equation in which you have that the velocity of the front is related to the square to the square of time, you have that it appears the square of time, but you have another terms that appear in this equation. So here you have the friction factor against the aspect ratio. This means the ratio between the lateral sides of the channels. Here you have the contact angle against aspect ratio, and here you have the contact angle against the relative roughness of the wall. You have considered smooth lateral wall and rough lateral walls. These are experimental values. Here you see that the roughness of the wall doesn't change too much the friction factor. On the contrary, the contact angle is largely modified by the relative roughness. So the main aspect here is that if this is the square of the length of the filling of the channel, these are experimental against time, and this is experiments are with thermals, and these are the results that you should obtain when you use Lucas-Washburn equations. On the contrary, when you correct with the friction factor and the effective contact angle, you obtain results like this, in which you can the results between experiments and analytical results are in very good agreement. But you may these results are described in this publication of last year. Well, once we have got a good description of the filling of the channels, well, we try to incorporate it to a code, so we can design not only a single channel, but a network of channels. And to do that, what we one of the objectives was to try to arrive to equations, or to arrive to conditions to chips in which we can govern the flow rate against time, and also the liquid, or the volume of liquid against time. So if you want to go through 3D codes, this would be too heavy, and you cannot undertake this kind of approach when you want to proceed to optimization for optimization purposes. So it is necessary to develop 1D codes. And when we started it, there was this code was not available. And what we have next done is to develop this kind of code in an environmental of MATLAB in what is called Simulink. This is quite graphical, and this can be used for anyone that, even if you are not a CFD fluid dynamics specialist, this is graphical system. So I explain a little bit this. More details are also in this paper. What you have here is, let's assume that you are interested in reproducing the dynamics of filling of this geometry of channels. So you have three branches here. The first one, the second one, and the third one. So first, what you have to do is to describe the fluid you are using, and you will have for each of these branches you will have a module, and you have to link this module. The module 1 is linked to module 2 and 3. Module 1 is linked to module 2 and 3, and this is done directly in a graphical system. So for this kind of problem, we have developed a code in which you can graphically add modules and construct the network in which you are interested in, and this will or this should reproduce the dynamics of the filling of the different branches. So this kind of model is, as I repeat, is quite easy to use. You have the graphical environment is quite easy to use, but you have a lot of equations behind this that you don't know, you don't need to manage or to handle. This is already incorporated in the system. So let's first see how, if this code is okay. And for that, what we do is we proceed to validation. So you can validate a code either with numerical or with analytical expressions, or you can validate it with experiments. So first, we validate it with analytical expression. So if you have this kind of problem, you have an analytical solution for this kind of problem. So this is quite nice because you can see if a long time you obtain the same results. So we have three phases here. The phase A is in which you have only the first channel that is filled. The phase B is that you started, the channel 1 is already filled, and you start to fill the channel 2 and channel 3. And the phase C is the case in which the flow has, the channel 2 has been filled also, and you have the channel 3 that is filling. So this is the dynamics of the branch 1, this one, and so at the first time, this is filling, and then it stopped because it's already filled. This is the branch 2 and branch 3, both of them as the geometry is symmetric. Both of them are coincident until the branch 2 it's filled and it's stopped. This is the phase B. And finally, you have the phase C, in which this branch is filling. So here you have with samples, you have the results of the numerical results, and with full lines, you have the analytical results. As you see, they are both coincident. So here everyone is pretty glad because you can validate the code with a simple geometry and for which you have analytical solution, and it seems that the code is working fine. But we have validated the code also with experiments in a more complex geometry. And here we have a T. This is the experimental results, and this is the numerical animation what is predicted by the code. So at the left, you have the experiments, and at the right, you have the numerical results. So as you see, well, results are in quite good agreement. So we are quite, well, the code is quite confident for us, and we think that we can use for the purposes that is to optimize the network design with towards some specific objective. So what it follows is we adopt a network configuration, and the filling, the liquid filling of the network depends on the set of channel width and lengths adopted. So this with the channel width and the length of these channels will be the input of the machine learning algorithm that we will use. We will use in this case. So you have individuals. Each individual is formed by the set of channel width and length that you have adopted.
Guillermo, I'm sorry to interrupt you, but could we just speed up a little bit, because it's now like 1 hour, just to have time for the questions, if it's okay with you.
I'm sorry.
Okay.
I will finish in a few minutes.
Okay,
so,
great.
The main aspect is that you have individuals, and you have a minimization process that you have to obtain which is the best condition for this, and this is done with a machine learning genetic algorithm. So for this kind of geometry, in this case we have adopted this kind of geometry, and what we wish to obtain is we wish to fill or to have some volume in a given time. So this is the channel configuration that we have adopted, and at the end you obtain different sizes of channels, and here, and this is the in dots you have the experimental results, and with the symbols, with the line, the broken line, you have the numerical results, and the same here. So the system, with this system you can predict correctly the volume against time. In this case, instead of having a linear volume increase along time, the second case is a case in which we have a parabolic volume that is a long time, that is being filled, and again we have experiments and numerical results. And in this case, what we have changed is that we have also accepted to vary the length and the size of the size of the channels. So also we have developed some strategies to avoid the asymmetries, and to take into account the elbows and curves. And well, as we have been working during some time biomimetic inspired flow control, I passed this one, but we have worked with leeches, the swing of leeches, the flapping of foils related to the flying, and this is related to the glottal flows a long time. We have now, or we are interested now in the design of a channel configuration of channels related to leaf venation, in which we are trying to obtain some information or some criteria to develop configuration of channels inspired on leaf venation. This is unpublished, and this is ongoing work. And well, finally, I arrived to conclusions. Chip design with specific goals require the use of optimization techniques. This is more crucial in capillary microfluidics, where chips require large effort of engineering design. Capillary chips are particularly adequate when it is required to control the liver of low volumes or low shear stress, and when using rather viscous or non-Newtonian fluids. And we developed a strategy to obtain chips in which we control the flow or the rate or the liquid against stand. Even non-explosive, main applications are related to diagnosis, and well, present works are related to dynamic aspects of capillary filling, either to separation, classification, or mixing, and also well to the design of configuration of them. Well, that's all, and thank you very much, and sorry because I exceeded a little bit on time.
Thank you very much, Guillermo. It was a very, very interesting presentation. Just a second, I'll stop the recording.